So, good afternoon, everyone. Let's start the GI tract from the esophagus to the small intestine. Okay, so our first slide represents the esophagus. So let's see it. On the slide, we can find two sections of the esophagus. The first represents the upper portion, the other the lower portion of the esophagus. It eventually shows different arrangement of the muscle cells and the types of the muscle cells, because the striated muscle fibers are the predominated ones in the upper portion. Eventually it's really close to the esophagus, so and the pharynx and the oral cavity. So this portion is under voluntary control, while the lower portion represents mainly smooth muscle fibers under involuntary control. Now, let's see the upper part first. So when we just describe a luminar organ, you remember we can distinguish different layers of the luminar wall. So we have mucosa, we have submucosa, we have muscularis externa, and adventitious serosa. Please switch off your microphones. So the tunica mucosa is the first layer. So this darkly stained thick area represents the typical layers of the stratified non-keratinized squamous epithelium. So here we have the stratum basale cells, then stratum spinosum or polygonale cells, and stratum planocellulare. So it's a typical stratified non-keratinized squamous epithelium. We can find some invaginated portions of the connective tissue making papillae. So here we have some vessels related to the lower portion, because it's not a flat, not a completely flat surface. So the invaginated connective tissue under the epithelium invaginates into the upper parts. Then here we can find there is a muscle layer. Here we have glands, everything. So immediately underneath the epithelium, we have the lamina propria. So this is the lamina propria, which is eventually moderately dense connective tissue. Not so many gaps are presented among the cellular elements and the connective tissue fibers. So this is the lamina propria, and definitely the majority of the cells are lymphoid cells, mainly lymphocytes. Underneath the lamina propria, we can find this area. This is a really thick muscularis mucosae. So there is a muscle layer which is eventually represents smooth muscle fibers in longitudinal appearance, in longitudinal arrangement. So there is a really thick tunica muscularis mucosae, lamina muscularis mucosae. So these are the layers of the mucosa. Here we have the epithelium mucosae, which is stratified squamous non-keratinized epithelium. That is the lamina propria, which is moderately dense connective tissue with lymphoid elements. And then we can see the muscularis mucosae, which is a thick smooth muscle layer representing longitudinal smooth muscle fibers. So underneath the muscularis mucosae, we can find the submucosa. So just look at that. So this area, this is the submucosa, which is eventually more looser than the lamina propria. We have numerous gaps among the fibers. We have glands, excretory ducts. So looking at this, there is a mixed gland. Here we have the typical mucus acini with some serous components. There is a duct. By the way, here we have a line by stratified cuboidal epithelium. Many vessels we have there and there, here as well. And definitely the main components of the submucosa are the collagen fibers. So these coarse pinkish bundles are the collagen fibers and representing fibrocytes. You know, the fibrocytes secrete them. So eventually they are the main components of the submucosa. Yeah, and call these glands, these mixed glands, mainly mucus glands called esophageal glands, but they are just simple mucus glands, mainly mucus glands. Then next layer, when we just decrease the magnification, we can see the layers of the tunica muscularis. The tunica muscularis, two layers of the smooth muscle layers. We have a inner circular and outer longitudinal muscle fibers, smooth muscle fibers. But when we just zoom in, we can see the typical appearance of the striated muscles. So beautifully seen the striation, the nuclei are at the periphery underneath the cell membrane. They are multinucleated cells. This is the inner circular, and then we have the outer longitudinal fibers. This is also striated muscle fiber, multinucleated nuclei are underneath the periphery. But I wanna show this as well. So look at that. This is smooth muscle. So it's smooth muscle arranged. Eventually, it's also the outer longitudinal appearance of the muscle fibers, but it's smooth muscle. So the nuclei are at the center of the cells. It's only one nucleus presented in each. So this is a typical smooth muscle appearance. But when we just decrease the magnification, we can see the majority of the cells, the muscle cells, are striated muscle cells, and we have smooth muscle cells as well. So I said at the upper part of the esophagus represents mainly striated muscles because of the continuation of the pharynx and, you know, under the oral cavity. So this part has more effect on the so more voluntary under more voluntary control than the lower portion. That's the reason of the arrangement of the muscle types, muscle fibers. So the predominated components of the muscles are striated muscles. And then just when we are looking at the external surface of it, we can see loose connective tissue. Is a typical loose connective tissue having blood vessels, adipocytes, and definitely brown adipocytes. So this is the typical appearance of the brown adipose cells. So the multilocular fat droplets are presented in the cell, not in the cytoplasm, because the cytoplasm is pushed to the cell membrane with the nucleus as well. So many fat droplets are presented in one cell, and among the brown adipocytes we can find some white adipose cells as well. You know, the brown adipose tissue for the heat production, while the white adipose tissue for the energy storage. So it's a typical adventitia, that's the outermost layer. You know, just the last part of the esophagus, approximately it's 2 centimeters long, wears peritoneal covering, so only the lower portion of the esophagus represents serosa. In the neck region, in the chest cavity, the esophagus surrounded by loose connective tissue. Therefore, the outermost layer of the luminar wall is loose connective tissue called adventitia. Let's see the difference with the lower part. It's look at that. The epithelium is the same, so stratified squamous non-keratinized epithelium. That's the same. So the stratum basale, stratum spinosum, and planocellulare. Maybe this area is a little bit better. So this is the lamina propria, which is moderately dense connective tissue. Collagen fibers are presented with lymphocytes and numerous other lymphoid elements. Here we have a lymphoid aggregation. Just look at that. There is a lymphoid aggregation. And under the lamina propria, we can find the muscularis mucosae. This is a really nice distinct layer having smooth muscle cells in longitudinal arrangement. So this is the muscularis mucosae. So from there to there is the exact thickness of the mucosa. Then underneath the mucosa, we can find the submucosa. This is it. Having collagen fibers, fibrocytes. These are the fibrocytes, the nuclei of them. And definitely vessels. There are also many, many, many vessels we have. And underneath, we can see the two layers of the muscularis externa. This is the inner circular, that is the outer longitudinal, and we just when we just zoom in, we can see it. It's a smooth muscle, almost completely smooth muscle, fibers everywhere. Let's see the outer layer also. This is the typical outer circular layer. The nuclei are at the center. They are not multinucleated cells. So I think I cannot see striated muscle fibers, but what I can see, it's really typical separating the two muscle layers, the inner circular and outer longitudinal in the GI tract, because between the muscle layers, we can frequently find a part of the enteric nervous system called mesenteric plexus or Auerbach plexus. So just look at that. So mesenteric, also called Auerbach plexus. It's part of the, so this area, this is it. It's part of the enteric nervous system, which is independent from the central nervous system. That means, for example, the patients in the condition of brain death still represents peristalsis because of the enteric nervous system. And the Auerbach plexus or mesenteric plexus is typically located between the two layers of the muscularis externa. It represents ganglionic cells, parasympathetic ganglionic cells there, and unmyelinated nerve endings, nerve fibers. So this is a typical Auerbach plexus or mesenteric plexus. These plexuses are presented only in the muscularis externa layer.
Dr. Katz,
yes.
Can you repeat again? They represent parasympathetic gangliotic cells and?
Parasympathetic ganglionic cells, these are the darkly stained ones. And these are the nerve fibers, the unmyelinated nerve fibers.
Okay, thank you.
Welcome. And here we have another. this. And here maybe we can see the nucleus of the ganglionic cell, just the shadow of that, the silhouette of that. So yes, maybe there is another. Yeah, here we have the ganglionic cell, and this is the whole mesenteric plexus. Okay, let's see the surface. It seems a really sharp covering here at the surface. That means, so I'll try to show some nuclei, but I can't see. But it's really sharp, the exact border. So we can see the peritoneal covering. This is that represents the lower portion of the esophagus when the abdominal cavity covered by serosa. So the outermost layer in the abdominal cavity is the serosal covering, which means the mesothelial cells cover the external surface, the external side of the organ. So look at that there. It's completely different than the previous one when we just went through the external surface and there was a messy appearance. So here numerous fibers, fat, everything. So there is no sharp border, no sharp edge of the layers. Okay, that was the esophagus. Let's see the stomach. That's the number 62. Yeah, that's the one. This is the number 62. So at the inner surface of the stomach represents numerous gastric glands lined by gastric mucosae. So the gastric mucosa, the epithelium of the stomach, is simple columnar epithelium. So just look at that. So this area here we have columnar cells covering the inner entire surface of the stomach. These are simple columnar cells, and these columnar surface regularly represents funnel-shaped invaginations called gastric pits. These are the entrances of the gastric glands. So these are the gastric pits lined by simple columnar epithelium as well. But they are, I wanted to say an important stuff. So when we just raise the magnification, these cells are a little bit similar to the goblet cells, but they are a little bit stained darkly than the goblet cells. They are really bright, but just looking at this area, they maybe look like goblet cells. No goblet cells are presented in the stomach. These are typical mucus secreting cells, but no goblet cells. You know, the goblet cells are unicellular glands, but these cells are the typical mucus secreting simple columnar cells of the stomach. So no goblet cells inside.
Okay.
Yes?
What they called gastric pits?
These are the gastric pits, the funnel-shaped entrance.
Okay, thank you.
Okay, so maybe it's a good one. So look at that. This is the gastric pit, and underneath this area represents the gastric gland. So we can find the gastric gland. So this is the gastric pit. That's the entrance of the gastric gland, and then there is a tubular gland going down to the muscularis mucosae. So there is a long tube-like something having different parts. The exact beginning after the funnel-shaped pit, this area is just called isthmus. This is the isthmus, where the lumen of the gastric gland dramatically decreased. And then we can find the neck region down, and then we can find the basal part, or just called base of the gland. So base, here we have the neck, and there is the isthmus, approximately. So gastric pit, this is the isthmus, neck region, and base. In the gastric glands, we can find 5 types of cells. We have hormone-producing cells, we have enzyme-producing cells, and other type of factors producing cells, and some undifferentiating cells. From these 5 types, we can identify 3 types of cells because the enteroendocrine cells are not well presented. They are located mainly at the bottom, and they are really similar to the main type of cells of gastric glands. So it's impossible to find enteroendocrine cells. Mainly the immunocytochemistry can reveal them. And the undifferentiating cells are also rarely seen. If we can see cells showing the typical signs of the mitosis, we can see sometimes, but they are rarely seen. So undifferentiating cells eventually providing sources for the cells in the gastric glands are rarely seen. So what we can see, the chief cells are located at the bottom of at the basal part of the gastric glands. So these darkly stained areas represent mainly chief cells. So these are the chief cells. Chief cells secrete pepsinogen. The pepsinogen, is a typical enzyme for the digestion of the proteins. So the pepsinogen is secreted by the chief cells. Pepsinogen, we just say gene at the end of the word, because that means it's secreted by an inactive form. So the inactive pepsin, called pepsinogen, secreted by the chief cells. Next type of cells, the parietal cells. Parietal cells are the lightly or eosinophilic structures there, a well-presented nucleus. Sometimes we can see it's similar to the scrambled eggs. So here we have the exact egg, and there is the cytoplasm, the pinkish cytoplasm. So more ovoid, much more bigger cytoplasm. These are the parietal cells. Parietal cells secrete hydrochloric acid. The pH in the lumen of the stomach is approximately 2 to 4, but the average is really close to the 2. So it's a really acidic, caused by the hydrochloric acid secreted by the parietal cells. Because the enzymes in the stomach juice can work on lower pH, so on acidic pH. That's the reason why we have hydrochloric acid secreting cells in the stomach. And beside the hydrochloric acid, it secretes a small glycoprotein called intrinsic factor. Intrinsic factor, it's a small glycoprotein binding the vitamin B12. The intrinsic factor just binds this vitamin B12 from the foods, and in the intestine only with this intrinsic factor can help the absorption of the vitamin. So without the intrinsic factors, we cannot absorb vitamin B12 from the foods. This vitamin is essential for the erythropoiesis. The lack of vitamin B12 causes anemia. So this anemia, called pernicious anemia, caused by the lack of vitamin B12. So this cell eventually essential for the erythropoiesis. And you know, when if the patient has cancer or in obese persons, we can eventually do gastrectomy when we remove the majority of the stomach. We should support, or we should regularly check the vitamin B12 level in the blood because it's essential for the erythropoiesis, and sometimes we can inject this vitamin into the patients just as a supplement of the vitamin B12. So parietal cells, moving a little bit up, so we can find more parietal cells closer to the surface there. So it's really nice. These are the parietal cells. Many over the surface or closer to the surface secrete hydrochloric acid and intrinsic factor, which is essential for the absorption of the vitamin B12. That was the second type, what we can identify. The third type is a simple columnar, simple cuboidal cell close to the neck region, therefore called it just mucus secreting neck cells. So these cells here, these are mucus secreting neck cells. At the neck region, obviously, they are also secrete mucus just for the inner surface of the stomach. The mucus layer in the stomach lumen is approximately half centimeter thick. So there is an extremely thick mucus protecting the layers underneath the lumen, because immediately under the mucosal, this mucus barrier, the pH is close to the neutral, so it's 7. Because this is the normal environment for the cells in the gastric glands. But in the lumen, we have a really acidic pH. So eventually the integrity of this mucosal barrier, this mucus barrier is essential. By the way, if the mucosal barrier or mucosal layer disrupted, there is a trigger factor for the ulcers. So can cause some ulcers. By the way, the alcohol intakes also can triggers the disruption of this mucus layer. So especially the shots, so the vodka, whiskey, so this kind of alcohols containing high level of ethanol directly harmful for the mucus barrier. Okay, so again, gastric pit, isthmus, neck region, basal part, and then at the bottom and among the gastric glands we can find eventually loose connective tissue having numerous lymphocytes. This is the lamina propria. So this is the lamina propria just among and underneath the gastric glands. Under the gastric lamina propria, we can find the muscularis mucosae. This is it. This thin layer is, this layer eventually represents inner circular, outer longitudinal muscle fibers, but rarely seen the different arrangement between the fibers, maybe here, maybe here. So the inner circular, outer longitudinal layers. And then under the mucosa, you know, that was the mucosa. So under the mucosa, we can see the submucosa. This is a little bit destroyed. It's artificial. Loose connective tissue we have, having adipocytes, blood vessels, connective tissue fibers, and also we can find a typical part components of the enteric nervous system called Meissner's plexus or submucosal plexus. Here we have this. That's the one. It's quite similar to the myenteric plexus, having unmyelinated nerve fibers and ganglionic, parasympathetic ganglionic cells. The Meissner's plexus, I try to find a better one. These are vessels here. These are also, but I can see the ganglionic cell. The number of the Meissner plexus is much more fewer than the myenteric. That's the reason why we can see it's partly just a half of the myenteric is presented. No, it's not. Here. This is it. So this is it. It's a little bit better. So Meissner's plexus. So Meissner's plexus having parasympathetic ganglionic cells, but here we can see the ganglion, exactly just the nerve fibers. So Meissner's plexus also part of the enteric nervous system and innervates the vessels, the wall of the vessels in the submucosa, and also innervates the muscularis mucosae. So eventually the movements of the mucosa. Okay, let's move on. Next layer. Next layer, muscularis externa layer. Here maybe we can see the 3 layers. The stomach represents three layers in the muscularis externa. Eventually the arrangement is the opposite than the arrangement of the muscle fibers in the heart ventricles. You remember the myocardium at the level of ventricles built up by the outer oblique, middle circular, and inner longitudinal. So the opposite is here. So here we have the inner oblique, middle circular, and outer longitudinal layer of the smooth muscle. So the inner oblique, middle circular, outer longitudinal layers of the muscle fibers, smooth muscle fibers, and among the muscle fibers we can find the other type of the enteric nervous system. Here these are the Auerbach plexuses, the myenteric plexuses, innervating the muscularis externa. And then this thin layer, this everywhere, is the serosa, because the stomach is intraperitoneal, completely covered by the peritoneum, and at the outermost surface we can find the mesothelial cells, and there is a really thin range of for the fine reticular fibers and connective tissue fibers called submucosa. This is the submucosal layer. So this is the subserosa. This is the serosa covering, having mesothelial cells, and this is the subserosa. Okay, that was the stomach. let's move on and see the cardia, the 63. So, you know, the cardia classification belongs to the stomach. Therefore, it's a muscular ring, just letting the food bolus into the stomach. But also represents parts. The upper part of the cardia represents the lining epithelium of the esophagus. So this is a typical section of the cardia. So when we just looking at the surface here, we can find the typical appearance of the gastric mucosa. These are the gastric pits having simple columnar epithelium. This is the basal part. These are the chief cells secreting pepsinogen. Here we have the scrambled eggs. They are the parietal cells secreting hydrochloric acid and intrinsic factors. And that the neck region, here we can see some cuboidal cells. They are the mucus secreting neck cells. So it's a typical stomach mucosa. Here as well. So we can see everywhere the gastric pits. There is also it's a rugae, you know, the folds of the submucosa. Then we can find the area which is completely different than the previous ones. So look at that. This is a stratified non-keratinized squamous epithelium. Here we have the stratum basale, stratum spinosum, planocellularis cells, but this is a transition. Look at that. Here we have it gradually changes. This is the exact level where the epithelium becomes simple columnar. So here we can see the portion of the esophagus. This is the cardia. This is the esophagoventricular junction. So here we have, there's the other side where the gastric mucosa begins. So there is the esophagus. So that slide represents the cardia level. Let's move on. Let's see the duodenum, the 65. Duodenum is the first part of the small intestine. In the small intestine, we can mention 3 important surface increasing structures, such as the Kerr-Kring folds. They are the plicae circulares. These big, huge submucosal projections just increasing the surface, called Kerr-Kring folds. No. So Kerr-Kring folds, these are the Kerr-Kring folds. And onto the surface of the Kerr-Kring folds, we can find these finger-like processes called intestinal villi. These are the intestinal villi. That's the second type of surface increasing structure in the small intestine. And onto the top of the villi, we have cells having microvilli. That's the third one. In the small intestine, we have 3 surface increasing structures. Kerr-Kring folds, they are the projections of the submucosa. Then intestinal villi, the finger-like processes, they are the projections of the lamina propria. And onto the surface of the villi, we have cells having microvilli, and that's the third one. So Kerr-Kring folds, villi, microvilli. These are the surface increasing structures. So let's see the here the surface. So this is a villi. These are the villi, many. And look at that. We can easily identify the goblet cells. So the typical appearance of the unicellular goblet cells presented. And these cells, these are simple columnar cells. It's a simple columnar covering. And this something, just look at that on the opposite side, and there is a silhouette of the mass of microvilli. So we can see the smallest surface increasing structures. So microvilli there. The cells, the simple columnar cells, bear another name called enterocytes. So they are the enterocytes. Enterocytes line the entire surface of the intestines, from the small to the large intestine. So this simple columnar lining epithelium, also called enterocytes. Then inside, you remember I said the villi represents part of the lamina propria. So the lamina propria, it's a cellular rich loose connective tissue. You can see numerous cellular elements. By the way, when we increase the magnification, we can see numerous plasma cells. These are these are plasma cells. Here we can see. Look at that, beautiful. The huge clumps of the chromatin at the periphery. So that is a typical clock face or cartwheel-like manner of the chromatin. So these are plasma cells. So lamina propria is cellular, rich, loose connective tissue with numerous cellular, mainly lymphoid elements. Yeah. And also in the lamina propria, we can find these circular units. These are glands. These are intestinal glands called Lieberkühn crypts. These are the Lieberkühn crypts. So crypts of Lieberkühn or Lieberkühn crypts are the intestinal glands secreting digestive juice into the lumen. And the crypts of Lieberkühn built up by mainly enterocytes and goblet cells. But among them, we can find some other cell types as well. So let's see these cells. First, this is a good aspect. Here we have the simple columnar cells, the enterocytes, and these are the goblet cells. But I'm showing another one, this. So some of the cells are different. So these cells represent these pinkish granules inside. These are lysosome granules. The lysosome, so lysosome is an antibacterial substance just against the fungi, bacteria, and other bugs in the intestine. But beside the lysosome, we have another molecules as well, and these granules just fight for the foreign bacteria, foreign viruses, and the cells called Paneth cells. So the Paneth cells secrete this enzymatic fluid. So Paneth cells secrete the lysosome. This is really typical for the small intestine, the Paneth cells, the presence of Paneth cells in the Lieberkühn crypts. not just Ponnet cells are presented. So the Ponnet cells release their granules into the lumen of the crypts of Lieberkühn, and eventually these glands, these Lieberkühn crypts, just eventually pushes their secretum into the bottom between the surfaces of two villi. So eventually the ducts of the crypts of Lieberkühn just conveys the juice between the villi. So we have another cell type, which is a little bit, that's presented, a little bit different than the previous ones. So looking at this, here we have the simple columnar goblet cell, Ponnet cells, but just looking at this and that, at the bottom of the cell, we can find some reddish something. This is different type of cell called enteroendocrine cell. You know, they secrete hormones, endocrine and paracrine hormones, and the bloodstream or the interstitial fluid carries it toward the targets. So eventually they are the enteroendocrine cells, and eventually it's easy to identify here in the crypts of Lieberkühn because they accumulate their secretum at the bottom of the cells, while the Ponnet cells represents the material at the apical part of the cytoplasm, the enteroendocrine cells at the bottom. So and beside of them, so the simple columnar enterocytes, goblet cells, Ponnet cells, and enteroendocrine cells, we can find sometimes undifferentiating cells. They are the sources of the different cell types in the crypts of Lieberkühn. Okay. Yeah, they were the typical components of the lamina propria. Let's see the muscularis mucosae. It's a thin layer which is represents inner circular, outer longitudinal, smooth muscle layer. So this is the muscularis mucosae. Let's see the submucosa. The submucosa of the duodenum is extremely unique because just looking at this area, it's easy to identify. We can see loose connective tissue. So it's a loose connective tissue having blood vessels, connective tissue, fibers, cellular elements, many capillaries, everything. But here, for example, we can find some glands. These are typical glands of the duodenal submucosa called Brunner's glands. The Brunner's glands, they secrete alkaline juice into the duodenum. So Brunner's glands, these are only the duodenum represents Brunner's glands, so only the duodenum represents glands in the submucosa. Therefore, the Brunner's glands also called submucosal glands. So this is a really good help to identify the exact slide because in the submucosa we can find these glands secreting alkaline juice, so it's a high content of bicarbonate, changing the pH in the duodenum. You know, the acidic pH content comes from the stomach through the pylorus, but the majority, so the really big majority of the pancreatic enzymes are able to work only on neutral pH. Therefore, the duodenal glands secrete this alkaline fluid, this juice, to change the pH into neutral. Then I just want to show this as well. So beautifully seen. It's a really good aspect about the Meissner's plexus. So in the submucosa, we can see the Meissner's plexus. This is a submucosal plexus having large ganglionic cells, parasympathetic ganglionic cells. These are the prominent nucleoli. These as well, so ganglionic cells and unmyelinating nerve fibers. These innervate the vessels in the submucosa and the muscularis mucosae. So they are part of the enteric nervous system. I just wanted to show this as well. So when we started with the Kerkring's folds, we can see numerous glands, Brunner's glands in the submucosa here as well. Okay, so the presence of Brunner's glands here, there are quite typical in the duodenum. Now let's move on and let's see the muscularis externa. So this is the muscularis externa layer, having two layers, inner circular, outer longitudinal, and among the muscle fibers. So this is the typical appearance. We can find Auerbach plexus is the mesenteric plexus. So inner circular, outer longitudinal layer of the smooth muscle. We can find Auerbach plexuses. They innervate the muscularis externa. And the outermost layer of the duodenum could be either serosa or adventitia, because everybody knows, I think, the superior part of the duodenum is intraperitoneal, while the descending inferior horizontal and ascending parts are retroperitoneal structures. Therefore, the retroperitoneals represent adventitia, and the superior part covered by serosa. So when we just look the surface here, it's a really sharp border. So this is the serosal covering, having mesothelial cells. But I think a little bit higher, yes, there we cannot see the serosa covering, just the typical view of the loose connective tissue. So this is the adventitia. So only the superior part of the duodenum represents mesothelial covering. So there, and there is a subserosa, a thin layer of subserosa. Okay, then let's see our other slide. We see the duodenum, but we have a section about the pylorus as well. That's the 64. Yeah. So this is the pylorus represents the typical surface of the stomach and the small intestine, and it's also formed by a huge, strong sphincter, smooth muscle sphincter. Here we have the pyloric sphincter muscle, smooth muscle fibers in various directions. So this bulk is eventually the pyloric sphincter. And on the small magnification, just looking at the surface or underneath the structures, we can see these numerous islands here. These are the Brunner's glands. Therefore, this is the small intestinal portion, and that is the stomach portion. And when we just look at the surface, so there, these are the typical. So this is the typical view of the gastric pits. So beautiful scene. And then, obviously, we have a transitional part. Here we can see the intestinal villi. We can see Kerkring folds, obviously because of the sphincter. So no typical submucosal folds, but villi are presented. So this is the duodenal surface, and here we can see the Brunner's gland in the submucosa. But look at that, it's really weird because there is darkly stained areas are a little bit similar to the basal parts of the gastric mucosa, but they are Lieberkühn crypts, just a little bit darkly stained, and numerous Paneth cells are presented here. So these cells representing these eosinophilic granules inside, they are the Paneth cells. Yes, there is a lymphoid infiltration. So in lymphoid infiltration, it's quite typical. It's not eventually weird because you know, the food and with the food we have numerous bacteria and fungi and viruses as well. So eventually they contacting these surfaces, contacting with these eventually dirty foods and liquids. So we need the help of the immune system. So that's the reason why we can find numerous immune cells and lymphoid aggregations. Then, so when we look at this surface, so the typical view of the gastric glands, but so here you see it's a bit weird because we can find some glands are really typical to the Brunner's glands. So this is the exact transition, by the way, here. You know, it's normally the Brunner's gland starts there, but some of the acini are presented here. So it's just a transition. But the epithelium is quite similar to the lining epithelium of the stomach. Yeah. Okay, so that was the pylorus. Do you have any questions? Everybody knows everything.
I have a question. When you zoom in to the upper part, there was some pinkish structures all the way to the top. Yeah, are those blood?
Blood, just
okay.
red blood cells.
Thank you.
Yeah. Other questions? Okay, so if you don't have more questions, we are gonna continue next week with the intestine, the small intestine and large one. So good luck for the midterm tomorrow and study.
Thank you, sir.
Thank you, sir.
You're welcome. You're
Bye.
welcome. Bye-bye.